Hello, everybody. Welcome to your free lesson. How are we all doing? Post a little hello in the chat so I can hear you. So not I can hear you. So I know that you can hear me, and I know that you can see me as well. I can see we've got Moonlight in. Well, I mean, Moonlight was here very, very early. I don't know if they're still here. Hello, Safa. Welcome to. How are we all doing today, then, my team? How is everybody doing? I'm interested to know how many of you have actually started school or college this week. I think quite often year 12s have like a little bit of a delayed start. Sometimes they might start in like a week's time or so. Zara, you started yesterday, unfortunately. Okay, there we go. Zara started yesterday. Jess, welcome Jess as well. Hello. And then also I'm going to put a little poll in the chat for you too. And very simply, have you already joined one of our courses? Yes or no? Using the poll in the chat. Have you been in any of our summer school lessons? Have you been in any of our main courses? I can see we've got about 50%, or maybe a bit more than that, a bit less than that actually, that have not been in any of our courses before. About 20% of you. Amazing there. Okay, so welcome team. Welcome everybody to our free lesson. Now, the whole idea of this is to give you a bit of a taster and a bit of an idea of what our live lessons are like here at My Ed Space. So if you've been on one of our courses before, then you'll probably know what our live lessons are like, but here's going to be a little bit of a taster of what year 12 is like, and particularly what joining an A-level lesson is going to be like. And then for those of you that have never been in any of our lessons before, welcome. And this is what all the chat is about. Now, those of you that don't know me, my name is Joe. I am Joe Does Biology. Do check me out on TikTok and Insta as well, particularly my A-level TikTok, where I break down all of the information that you need to know, okay? Now, I'm sure a big question that quite a lot of you probably have before we even start is exam boards. What exam board are we doing, okay? And here at My Headspace, we do courses for AQA and for OCR, okay? Now, some of you may be Edexcel, some of you may be Welsh Board, perhaps, and if you are, simple answer is most of the content is going to be suitable for you, okay? So even if you're Edexcel, nearly everything in AQA and OCR will be suitable for you, okay? Generally, if you are not one of these two exam boards, what I would recommend is to join OCR, because OCR covers slightly more. HMZ FX, no, this is not pre-recorded, this is live. I am speaking to you live right now. So those are the main exam boards that we do, okay? I do the OCR course. Laura, you might have come across Laura Does Biology. She does the AQA course. No worries, HMZFX, no worries at all. So a little bit about me, a little bit why should you come and learn with me, or why should you come and learn with Laura? We're both super experienced biology teachers. I used to be the head of biology at one of the top schools in the country, one of the top 10 schools in the country, and also used to be an examiner, and so did Laura as well. We both used to be examiners. And when it comes to A-level biology in particular, understanding the exam spec, understanding the mark schemes is really critically important. And because Laura and I were both examiners, we know exactly what you need to be writing and exactly what you need to learn as well. Laura is doing a year 13 lesson for you guys, HMZFX, okay? I think she's doing the year 13 one tomorrow, perhaps, which you guys would be welcome to join as well. Of course, you can join that. But for year 12, we're going to do a bit of year 12 content today, all right? Okay. Now, this is a free lesson. It is open to absolutely everybody. So get onto that URL, copy it, and send it to as many people as you possibly can. Let's see how many people we can get joining our stream before we finish today. So sharing is caring. Copy the URL, Snapchat it, Insta, whatever. Send it to as many people as you can. Let's see, and let's share the love. Let's see how many people we can get joining today. Okay, all right. Should we crack on? Should we make a bit of a start and kind of dip our toe into this world of A-level biology? Let's get going. So today I was thinking about what should I teach you guys in this free lesson, and what I thought about was DNA. Because DNA is probably one of the most important molecules when it comes to biology. And quite often we already have a bit of an idea of DNA as well. And some of you might have done triple science at GCSE. And if you did do triple science at GCSE, then you will have a little bit of knowledge of the structure of DNA as well. A really common question that people have is, oh, is it bad that I didn't do triple science? And no, it's not at all. Okay, so if you haven't done triple science, if you did combined science, then totally fine. You guys are still going to be able to smash your A levels. All right. So let's crack on. Where in the cell would you find DNA? Really easy question for us to begin with. Where would you find DNA? I can see we've got a nice answer from Safa as well. Where would you find DNA within the cell? Excellent there, Zara. Very nice. Any answers from Jess and Moonlight too? Yeah, nice there, Moonlight. It is our nucleus. Hello, Ada. Welcome. It is our nucleus. The nucleus is the kind of control center of the cell. It controls the activity of the cell, and it contains that DNA. Now, the structure of the DNA, there's a few different ways that we could describe this, but there's essentially kind of one key term that I'm looking for here, and later on in the lesson we're going to break down what we mean by that key term. I can see we've got Zara and Moonlight already popping some answers into the chat, which is fantastic. This is going to be about an hour, Safa. Okay, so it's going to be about an hour. We'll finish around about 5:30. Okay, it is a double helix. Exactly there, team. Very good. So the structure of DNA, we would describe it as a double helix. Now, is there something else that we could say as well? When you were doing this at GCSE, you probably would have said that it is a double helix made of... How could I finish that sentence? A double helix made of something. I can see Safa's coming in with the word polynucleotide. What might be a slightly more general term, perhaps? Polymer, fantastic there, Ada. A double helix made of polymer. And what I'm going to say is polymer strands. But how many polymer strands are we talking about here? How many polymer strands are necessary in order to form this double helix? I mean, the clue's kind of in the name, isn't it, really? It's kind of an easy one, this. How many polymer strands? Oh, not quite lots. I mean, I think maybe you're thinking about the number of monomers that make up a polymer, and there are lots of monomers to make up that polymer. Very nice there, Zara. It's 2. The double helix is made of 2 polymer strands. Exactly. Okay. So DNA. DNA is a molecule. DNA is just a chemical. Okay. So DNA is just a chemical, and it's a chemical that is a store of information. And later on, we're going to look at why it acts as a store of information. But before we can understand that, there's a couple of other keywords that I want to come back to that you would have looked at when you were doing your GCSEs. So what about a gene? What do we mean by the term gene? Just started already on polynucleotides. Exactly there, HMZFX. Well, I thought we could do the first lesson that you do in A-level biology, but the first lesson you do in A-level biology is usually quite boring. It's usually not that exciting. It's kind of cell structure or monomers and polymers. So I thought let's do something a little bit more interesting today. I can see we've got Zach saying a segment of DNA that acts as the basic unit of hereditary. Love that. Really nice definition. Zara, a small section of DNA. Safa, a small section of DNA as well. We've got this idea that it's a section of the DNA. So a small section of DNA. And when we think about like what is the purpose of this gene, this small section of DNA, the DNA is a kind of information store. It's the blueprint or the code, we could say, for the production of what? What is going to be produced from this DNA? A small section of DNA that codes for What does that small section of DNA code for? Post your answer into the chat. What do we think their team? HMZFX says a specific protein. Love that. You are absolutely correct. So the gene is going to code for the production of a protein or a sequence of amino acids. Nice, Moonlight. For a specific protein. And this is how the nucleus, this is how it is able to control the cell, because it controls what proteins are produced, and therefore the proteins are our molecular machines that go on to carry out all of the different reactions and functions within the cell. Okay. Now, later on in probably year 13, I'm going to take this definition and I'm going to pull this definition apart. Okay. But we won't talk about that until we get to year 13. And you'll come across that a lot in biology, that I'll give you a definition and then a few months later or next year or something, or if you go on to do biology at university, those definitions will start to pull them apart and be like, actually, there's exceptions to this, there's differences, it's more complicated, etc., etc. Okay? But this is what we're going to start with. So the gene is a small section of DNA. What do we mean by the term chromosome then? If the gene is a small section, what is the chromosome? That's a pretty sensational performance there, HMZFX, to go from a 4.4 up to an 8.7. Very, very well done. Not the section of a gene. Chromosomes, we're thinking slightly larger than a gene. Ada saying the DNA wrapped up together. Very nice. Or saying the DNA strand wrapped up together. Well, the chromosome is a length of DNA, and we're going to coil that DNA up. So in every single one of your cells in your nucleus, you have the full set of genes. You have your full genome within your nucleus. And it's about 2 meters worth of DNA. DNA is kind of like a string. About 2 meter length of DNA is wrapped up into the nucleus within every single one of your cells. So the nucleus is about kind of 1 micrometer across, so really, really condensing it down. And in order to do that, the DNA has to be coiled up. What is it made out of then, Moonlight? Do you mean the chromosome? Do you mean the DNA itself? What do you mean by your question there? So the chromosome, we would say, is a coiled structure of DNA. And the DNA is coiled up. Great. Yeah. Okay. So the chromosome itself. So the chromosome is made of DNA, Moonlight. What happens, and this is our first bit of A-level knowledge here, is the DNA, which is just the molecule, the DNA is wrapped around some proteins. And we call these proteins histones. Wrapped around histone proteins. And later on in year 13, we're going to take a look at these histone proteins, and we're going to talk about how the DNA being wrapped around these histone proteins affects kind of like the way that the DNA is expressed, the way that these genes are coded, and is one of the reasons why different cells carry out different functions and you have specialized cells in your body. HMZFX, you wouldn't be asked to draw the structure, no, but you might be asked to label the structure or identify some key parts to it. And later on, I've got some diagrams of the structures, and we will go through and label them all together. Okay, it's a good point though, guys. Don't forget that in the description of the video down below, you've got the link to the workbook, so you can access that workbook. You can download it. You can then annotate it and make kind of add comments in the same way that I am doing here as well. Okay, so these genes, these chromosomes, the DNA is wrapped around the histone proteins, and this, our next kind of key term, our next A-level key term, this is described as something called chromatin. Chromatin. I'm going to try and avoid throwing too many new words at you today, because A-level biology does have a lot of new words in it, and quite often it can be totally overwhelming. Great question, HMZFX. The chromatin is the DNA wrapped around the histone proteins. Okay, so if you imagine let me draw a little diagram for you. These little kind of green blobs, these are my histone proteins. These are my histone proteins here. And my DNA, if I get my DNA, let's go for a nice dark blue for my DNA. Well, the DNA is kind of wrapped around these histones. And you see how by wrapping it around these histones, I can have a really long length of DNA. I can have a really long length of that string, and it's all kind of condensed much, much closer together. And that way it will be able to fit within the nucleus. So this is our DNA here, and those green blobs, these green blobs represent our histones. Okay, excellent. All right, let's move on and let's start to think about these polymers in a bit more detail, because a polymer is a kind of large repeating molecule. What do we call the individual subunits of a polymer? No worries, HMZFX. Anytime you got a question, do ask it into the chat. Very nice. Any other answers from Ada as well? Moonlight. Who else we got in the chat today? We've got Jess and Zara and Safa. The subunits of these polymers. Hello there, Tan. These are our monomers. Very, very nice, team. Now, the monomers, does anybody know what the monomer of DNA is? Does anybody know? If DNA is a double helix made of 2 polymer strands, what do we call the monomer of DNA? What do we call the monomer of DNA? What do we think? Any thoughts? Give you a clue. Begins with an N. Our monomer of DNA is, not the amino acids. Moonlight, you're almost on the right lines there. It is a nucleotide. Nice, Zara and Safa. A nucleotide. Sometimes we might refer to this as a nucleic acid. So sometimes we might refer to it as a nucleic acid as well. Now, nucleotides, these are the monomers, and these monomers are made of 3 individual parts. And for A-level biology, you need to know the 3 parts of these nucleotides. The first thing is we're going to start with a sugar, okay? Now, the sugar, a little bit like this. I'm going to draw it for you. What shape have I drawn? How many carbons do you think are in this sugar? How many carbons do you think are in this sugar based on the shape of it? Based on the shape, how many carbons do you think are in this? 5. Good, Moonlight. Maybe I caused confusion by drawing my extra little line there. 5 carbons. We call this a pentose sugar. 5, as in pentagon, pentose sugar. Any textbooks that I'd recommend? HMZFX. If you're doing AQA, we've got our My Resources pages. You can check those out on our website, which are fantastic for AQA. OCR will be coming very, very soon. And then on top of that, I'll have a little think. For anything on top of that as well. Okay. So this is a pentose sugar because it has 5 carbons. And does anybody know what the D stands for? Does anybody know what the D stands for? No, Vanessa? No, if you're just starting in year 12, you don't need to know this at all. This is a little bit of a teaser of things that you're going to be learning in year 12, okay? What does the D stand for in DNA? Nice there, Ada. Yeah, deoxyribose. So this pentose sugar, this is actually deoxyribose. And what we would say is that it is a ribose there. And the reason it's a deoxyribose, I will explain in just a second. Okay, I'll explain on our next slide. Now, we've got 2 other parts to our DNA nucleotide. Our next part, which I'm going to draw up here. Let's go for green. This is what we call a phosphate group. This is a phosphate group. In the chat, what elements do you think you would find in a phosphate group? Pretty easy. What element would you find in a phosphate group? Not the base. We're going to come to the base in a minute there, Moonlight. Phosphate group, it is going to be, what elements do you think we would find in a phosphate group? It is going to be our phosphorus. So we would have a phosphorus atom. Now this is going beyond A level even. You don't actually need to know this, but the phosphate group is a P. Then we've got a little bit like that. It's a phosphorus and then 4 other oxygen atoms all associated with each other, okay? Phosphorus is a really, really important part of DNA. And the phosphorus is used in some other ways in something called ATP that we will look at in the main course. We don't really have time to look at ATP today. And then our third part here is, and I'm just going to draw this initially as a bit of a rectangle, okay? This is our bit of a rectangle. And this is what we would describe as our base. This is what we describe as our base. Specifically, I would refer to it as a nitrogenous base. If it's called a nitrogenous base, what elements do you think you would find in the nitrogenous base? Clues in the name, bit like phosphate. Nitrogenous, what elements would you find in a nitrogenous base? We would find nitrogen. Excellent there. Zara, Ada, Moonlight, Safa, Zach as well. Fantastic. Nitrogenous base. So DNA, the elements contained within DNA, well, we have the phosphate, we've got the nitrogen, but we've also got, because we've got this sugar, the sugar is an example of a carbohydrate, and carbohydrates contain carbon, hydrogen, and oxygen. So the DNA has the elements carbon, hydrogen, oxygen, nitrogen, and phosphorus. And this is important. And when we look at other examples of biological molecules, when we look at proteins in more detail, when we look at lipids in more detail, when we look at carbohydrates in more detail, we will kind of think about the different elements contained within those different biological molecules. Great. Okay, so our 3 parts of our nucleotides, that is our sugar, that is our phosphate group, and our nitrogenous base. And we could perhaps say here our pentose sugar, just to be specific. And our pentose sugar for DNA is deoxyribose. Okay? Does the phosphate vary? No, it does not, Moonlight. The phosphate is always going to stay the same. The thing that does vary is the nitrogenous base. Now, one of you mentioned this earlier. I can't quite remember who it was, but one of you mentioned this earlier about different types of bases. Now, this nitrogenous base, there are 4 types of nitrogenous base. Does anybody already know what those 4 types of nitrogenous base are? Does anybody know what those 4 types of nitrogenous base are? What do you think? Excellent there, Zara. Fantastic. A, T, C, and G. So A, T, C, and it's terribly written G. A, T, C, and G. And Zara is even showing off with the full naming them. So A stands for adenine, T stands for thymine, C stands for cytosine, and G stands for guanine. And these, you'll need to know the full names of these for your A levels, okay? So let's get to know them. Let's get to learn what the full names are. Now, we can also split these 4 bases into 2 groups. We've got something called purines and pyrimidines. Now, I'm not going to go into too much detail here because I don't want to cause too much confusion. Ooh, Moonlight coming in with the U. We're going to talk about U in just a second, okay? I'm not going to go into too much detail between the difference between purines and pyrimidines because I don't want to cause confusion. But what I am going to say is it's based on the structure of the base, okay? Now, the pyrimidines are more simple. The pyrimidines are just a kind of hexagonal ring. The elements bind together. The atoms bind together and form this hexagonal ring structure. The purines are a little bit more complicated than that. The purines, try and draw my diagram a bit more accurately, have a hexagonal ring and then also have a pentagonal ring associated with it as well. So the purines are slightly more complex. And we can split these 4 different nitrogenous bases into being a purine or a pyrimidine. Now, adenine is an example. I'm going to use my highlighter here. Let's highlight our purines in yellow. Adenine is an example of a purine. Guanine is also an example of a purine. And our pyrimidines, let's go for blue. Our pyrimidines here, thymine is a pyrimidine and cytosine is a pyrimidine. Now, the eagle-eyed among you might be noticing something going on here. You might notice something going on. What's significant about these bases? Does anybody know what's significant about these bases and how they perhaps join up with each other? Because remember, the double helix, it's made of 2 strands together, isn't it? It's 2 strands together. So these bases actually line up with each other. Does anybody know in what way they line up with each other? Those of you that did triple at GCSE might know this as well. Ada talking about complementary base pairing, Moonlight as well, and Ada too. Yeah, our complementary base pairing. That's exactly what we're going to call it. So complementary base pairing. And our complementary base pairing is the fact that adenine, A, binds with T, and C binds with G. Okay? And so what can we see there? Well, we can see that adenine is a purine, thymine is a pyrimidine. Guanine is a purine, cytosine is a pyrimidine. So what we always get is that purines bind with pyrimidines. The purines and the pyrimidines join together in our complementary base pairing. Okay? So, complementary base pairing is quite complicated. And I've represented some little kind of lines here for our complementary base pairing. What I have drawn, these are things called hydrogen bonds. Now, you might have come across hydrogen bonds when you were doing GCSE. Those of you that are doing A-level chemistry will get to know hydrogen bonds very, very well. But hydrogen bonds are a sort of very weak type of bond between molecules. And the different bases form different numbers of hydrogen bonds. So adenine and thymine form 2 hydrogen bonds. Cytosine and guanine form 3 hydrogen bonds. And that's what makes them complementary to each other. That's why they line up, and why adenine doesn't line up with guanine, because it doesn't form the right number of hydrogen bonds. So there we go. That's our complementary base pairing explained. So this is DNA. Now let's move on to RNA. If DNA stands for deoxyribose nucleic acid, what do you think RNA stands for? If DNA is deoxyribose, what might the RNA stand for? What do we think on the chat? Ooh, not the duplicated version. Okay, let's break it down. So just like any nucleotide, nice there, Dimeji and Ada as well. Any nucleotide is made of our, let's recap, pentose sugar. What are the other 2 parts to any nucleotide? Let's just recap for a second. So we've got our pentose sugar and we've got 2 other parts here to our nucleotide. What are the 2 other parts? It is our phosphate and our base. Fantastic, Zara. So we've got our pentose sugar. Then we have our phosphate. And we have our nitrogenous base again. I always think it's worth saying nitrogenous base. Very rarely do you have to say nitrogenous base in exams, but sometimes for the mark scheme, you do have to have said nitrogenous base. So it's better to kind of prepare yourself and just say it all the time, because that way you're never going to not get the mark. So let's look at our structure. Well, the first thing is we have our phosphate group. We've got our phosphate group. It's exactly the same as it is in DNA. So our phosphate group there. And the phosphate group is still bound to this pentose sugar, but this time we have a different pentose sugar. So I've still got my pentagon, whereas in DNA, it was just like that. Deoxy means without oxygen. Like if you deoxygenate something, you remove the oxygen from it. So deoxyribose, we've removed the oxygen. Ribose is exactly the same structure, except for one OH group here, one kind of alcohol functional group on that ribose. And what that does is that changes the properties of the ribose. It changes the chemical properties, which means it interacts in a different way. And so it's used for different functions. And later on in your A levels, you will learn all about the roles of RNA and how the RNA is used in different organisms, how it's used in viruses compared to bacteria, compared to eukaryotes, and how that RNA is also used within eukaryotes in different forms. Sometimes it's folded up, sometimes it's used as a straight chain, and if you're lucky, I might even start rabbiting on and talking all about something called the RNA hypothesis and how life first started on Earth. That's not actually part of the A-level spec, but I find it very interesting. So quite often I end up blathering on about it. Great question there, Safa. tRNA. tRNA is called transfer RNA, and it's a special type of this RNA that is used in eukaryotes in order to produce those sequences of amino acids. And it's part of a process called translation. It happens at the ribosome, the site of protein synthesis that you would have come across at GCSE. And that's something that you'll learn quite early on in year 12. So kind of OCR crew, we're going to be doing that in like October time, basically. We're going to be looking at the process of translation. All right, let's get into it. So the next part here is our nitrogenous base. So then we have our nitrogenous base again, and this time we've got our adenine, we've got our cytosine, we've got our guanine, but we have one slightly different one. We have something called uracil. So we've got uracil in place of thymine. There is no thymine in RNA. It is a slightly different structure, okay? Now, uracil is still an example of a pyrimidine, though. So now the complementary base pairing, I'm going to put CBP for our complementary base pairing. Our complementary base pairing is now going to be adenine with uracil and cytosine still with the guanine, okay? So uracil is still an example of a pyrimidine. So this is our pentose sugar. This is our ribose sugar. Why is the OH attached? Great question there, Vanessa. So this structure, if I was to draw this structure in more detail and kind of show you the atoms, what it actually is you've got an oxygen at the top there. That oxygen is bound to a carbon, which is bound to another carbon, which is bound to another carbon, which is bound to another carbon, and that carbon is then bound to another carbon up here. And this one has an OH group here. This one is bound to the nitrogenous base and then has a hydrogen here. This one has a hydrogen and an OH group, and this one has a hydrogen, and then this one has the phosphate group and 2 hydrogens as well. There we go. So that's kind of what the atomic structure. Yeah, but we would refer to it as a hydroxyl group there. So this OH we would refer to as a hydroxyl, although so often in exams we would just write OH, and if you write OH that's totally fine, but the technical term is hydroxyl as opposed to hydroxide, because the hydroxide would be when it's in its ionic form. And those of you doing A-level chemistry will learn all about how the hydroxyl group dissociates to become that ionic form there, okay? So in comparison, this sort of atomic structure that I've drawn up here, the deoxyribose is very simply oxygen, carbon, 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 and that carbon is bonded to the carbon up there. And we have the same hydrogens here, and OH, and then this one has a hydrogen as well, and this one has 2 hydrogens and the phosphate group, and then this one's attached to the nitrogenous base. But instead, this carbon just has a hydrogen here. This one, it doesn't have that OH group. So on the right hand side, this is our deoxyribose. On the left-hand side, this is our ribose, and it's that OH group. That's the only point of difference between them, the only point of difference. And as you will see later on through your A-levels, this really influences the roles of these different molecules and how they interact within different organisms. Would you need to know how to draw that, Zara? It depends. Are you AQA or OCR? If you're AQA, no, you don't need to know how to draw that. If you are OCR, I've got bad news for you. Yes, you will need to know how to draw that. But OCR crew, we're going to go through it in loads more detail later on in the main course. I will construct it for you in more detail. We will explain it in a lot more detail too. Okay. But if you're AQA, good news, you actually don't need to know. You actually don't need to learn the structure. But if you are AQA, you do need to know the difference in terms of whether or not it has that OH group. Okay. So these are our examples of nucleotides or nucleic acids. These are the monomers. These monomers are going to join together to form a polynucleotide. So poly as in many, nucleotides, simple as that. So we've got one nucleotide here, and that nucleotide is now going to be joined to another nucleotide. And what happens is we form this bond here. And this bond is really quite significant. This bond is quite special. This we would call a phospho, as in the phosphate group, because the phosphate group is involved. Di, because there's 2 parts to it. Ester. So in GCSE chemistry, totally misspelled the word ester there. In GCSE chemistry, you would have come across ester bonds and the structure of esters. This is called a phosphodiester bond because basically you've got 2 parts to it. There's 2 sides to that phosphate group, and both sides have an ester bond associated. So a phosphodiester bond. And all together, this chain of phosphodiester bonds forms, well, there's 2 parts to it. There's, what's this again? What's the kind of general kind of class or category of molecule that blue pentagon represents? What's the general class of that blue pentagon? What would we say for that? That blue pentagon, what does that represent within our nucleotide? If the blue circle is the phosphate, we've got our bases there. That blue pentagon, that is our pentose sugar. Fantastic, Zara. That's our pentose sugar, which means that this chain of phosphate and sugars, we're going to call it our sugar phosphate backbone. Sugar phosphate backbone. And this sugar phosphate backbone is really stable. And this is what can allow the DNA to act as a kind of storage molecule. It stays around for a really long time. Would you have to say pentosaccharide? It's not a pentosaccharide, Safa. It's still a well, it's actually not a monosaccharide because it's not by itself, but it's a single sugar. It's a pentose sugar though, as in 5 carbons. Later on the main course, we're going to talk all about what we mean by monosaccharides, disaccharides, trisaccharides, etc. Okay. Essentially, it's when you have 3 sugar molecules. A trisaccharide would be 3 sugar molecules all bound to each other. So this is our sugar phosphate backbone, and this is what forms our polynucleotides. And this is done by an enzyme, and it's a really important enzyme called DNA polymerase. So the DNA polymerase is going to form this chain. Or if we were producing RNA, it would be RNA polymerase. And this all takes place within the nucleus. So these enzymes, what they do is they form the phosphodiester bond. So if we're talking about something like DNA replication, the DNA polymerase is going to be producing new DNA molecules during DNA replication. And DNA replication is a really, really important part of biology, and we'll kind of break it down and look at it in a lot more detail later on in the main course. I love the way some of you guys are asking questions. So if you do have anything, if you think I've skipped over something, if there's something you don't understand, if you've got like a bonus question, then do post it into the chat. I particularly love it when people ask me questions that I don't know the answer to, because then I always like to go and learn more, because I love biology and I love learning more about biology. So if you think you've got a question that I might not know the answer to, post it into the chat and I'll do my best to try and answer it. All right, let's go on to how these polynucleotides arrange themselves for DNA. Well, we've got that double helix, haven't we? We can see that here. We've got our 2 strands. So this is our 2 polymer strands, as we mentioned earlier. And those 2 polymer strands, we have our base pair. And the important part is the pair there. Now, students use this term incorrectly They use this term incorrectly all the time. The base pair refers to the complementary pairing between nucleotides of different strands. So it's between the nucleotides of different strands. In response to your question there, Jess, do we get tested towards the start of year 12? At My Headspace, we don't formally test you. As part of the course, you will get access to kind of exam question homeworks, that really helpful practice, along with video solutions where I talk through all the answers for you. And then you will also get access to a monthly assessment to kind of track your own progress. But we don't have any formal assessments or anything like that. In school, you probably will. In school, you will probably have formal exams, probably 1 per term or something like that. And something like this will almost certainly come up in them because of just how important and how foundational it really is. And then we can see here we have our 2 sugar phosphate backbones, our 2 polynucleotides joined together by these hydrogen bonds as well. Great. So this is our structure of DNA. I'm not going to go into too much detail on the structure of RNA because I think that will make more sense when we have a better idea of where RNA is used, particularly within that process of protein synthesis. But essentially, RNA doesn't form the 2 strands. RNA is single-stranded and folds up on itself. And later on in the main course, I'll tell you all about that. Okay? So, one thing I want to leave you with, the last thing we're going to look at today relating to DNA is, well, how do we know? Like, how do we know all this? Like, okay, all right, Joe, cool story, bro. Like, well done, you've told me what the structure of DNA is. You could be lying. Like, you could be making it all up. How do I know that it's these little pentagons and these little circles and these little squares? Like, it doesn't make any sense. Well, the first thing we have to do is we have to extract the DNA. And how we actually know the structure of, like, how we know that the carbons are arranged in that way, and the structures and the fact that the nitrogenous bases are these hexagons and pentagons and stuff. How we know that is some really quite complicated chemistry. Those of you that are doing A-level chemistry will learn about how we determine those structures. Something called NMR and spectroscopy as well. You'll learn all about that in A-level chemistry. How the atoms are arranged, you kind of have to take my word for it. But if you're interested, I will explain about how we do that as well. But the first thing we need to do is actually extract the DNA. We need to get some DNA so that we can analyze it, so that we can study it. And this is something that you can do at home, okay? So I'm going to talk through how we extract DNA, and you can go on to learn about that. Is it a pentagon though? Sort of, Moonlight. Sort of. Like, the atoms, because of the way the angles that the bonds can form, because of the structure of the electrons within the atom, they do kind of form a pentagonal shape. Okay. So it's all three-dimensional. That's a really good question there, Moonlight. So often I'm going to show you two-dimensional images and two-dimensional diagrams, but in reality, it's always 3D. It's always three-dimensional. The DNA, it's a double helix. It's wrapped round itself. It's a clockwise spiral the whole time. Okay. So how do we extract DNA? Safa says from strawberries. Exactly. Maybe you've done this before. So the first thing to do is to homogenize the cells. This word homogenize is basically a posh word for break open. We're going to crush them. We're going to break them open. We might blend them. If you did this before at school, extracting DNA from strawberries, you probably smash the strawberries up in a plastic bag or something like that. Is this required practical for OCR? Yes, it is, Safa. For AQA, it's not actually one of the core practicals that you will be assessed on, but it's something you kind of also need to know for AQA. What we're then going to do is going to add some detergent. Now, the detergent there, what this does is this breaks down fats. It breaks down lipids, and it's going to break down the cell membrane. But also something called the nuclear membrane. Now, the nucleus is surrounded by a special membrane that holds the DNA in place. So it breaks down the cell and the nuclear membrane. And by doing that, the DNA is now kind of free. The sodium chloride is ionic. What this is going to do is this is going to kind of neutralize the charge on the DNA. Because of the phosphate group, the phosphate has a negative charge on it. And this means that the DNA molecule has a negative charge overall because of that phosphate group. So the sodium chloride there is going to neutralize the charge, and then we're going to add some water too. We're going to place it into a water bath, 60 degrees. That's going to kind of denature any of those histone proteins as well. So we can extract the DNA from those histones. And then we're going to place it into an isobath and also add protease enzymes. What do we think those protease enzymes are for? Why are the protease enzymes necessary? So sodium chloride, you're correct there, Moonlight. Sodium is positive, chloride is negative. So overall, sodium chloride is neutral, but the ions are separate, aren't they? The ions are not kind of, especially when it's in solution, the ions are free to move. So yeah, overall, it's neutral, but when those ions are free to move, the positive sodium ions will bind with the negative phosphate ions or phosphate groups of the DNA. So Zara and Ada talking about breaking down the proteins exactly, and those proteins. What are those proteins called associated with the DNA? So to break down, what did we call those proteins that the DNA is wrapped around? Those are our histone proteins. So breaking down the histone proteins. And then what we're going to do is we're going to add some cold ethanol, and the DNA will form a precipitate. And so you can do this really, really easily at home. Like, you don't have to add the protease enzymes, but you can get ethanol from anywhere. Sodium chloride is just salt. Detergent, you can just use fairy liquid. You can do this at home, and what you get is this little layer. And this little layer here is the DNA, and you can kind of wrap it around a little cotton bud or something. And there we go. That's how we extract DNA. All right. We've learned a lot today. We've gone through quite a lot. Let's have a little look at some questions. So the first one is a multiple choice question, and multiple choice questions at A-level are really quite tricky. So the following statements describe features of DNA structure. Which of the following statements is/are correct? Number 1, purine bases always pair with pyrimidine bases. Put a little T or F in the chat. True or false? Purine base with pair with pyrimidine. True or false in the chat? What do we think? Zara says true. Ada says true. Absolutely correct there. Yes. Remember, adenine binds with thymine. They are purines and pyrimidines. Number 2, adjacent nucleotides are joined by phosphodiester bonds. Again, true or false. Put a little T or F in the chat. What do we think? Any thoughts from Jess and Faisal, Demeji? Who else have we not heard from a little while? Zach as well. HMZFX, if you've finished your noodles by now. AV Edits. Are nucleotides, are they joined by those phosphodiester bonds? Yeah, nice there, Demeji. It is true, they are joined by phosphodiester bonds. And then the quantities of adenine and guanine are always the same. Again, true or false? What do we think? What do we think for this one? Adenine and guanine, are they always the same? Ada says false, Zara says false. Excellent, and Moonlight too. False, false, false. Yes, because adenine, the proportion of adenine will always be the same as the proportion of thymine. So if we have, 20% adenine, we will have 20% thymine, which means we will have 30% guanine and 30% cytosine. So these would be the same, but the adenine and guanine won't necessarily be the same. So our correct answer there, 1 and 2 are correct. So we are left with B. Now, when a cell divides, it produces an identical copy of its DNA during the process of DNA replication. Explain how base pairings ensures that the DNA copies produced are identical. 3 marks here. Let's break it down together. So the first thing we want to talk about is what is it about the DNA bases that leads to this kind of copying? What's kind of special about those DNA bases? What's that 3-word term that we would use here? Complementary, exactly, Zara. We would refer to it as complementary base pairing. So I'm going to start by saying complementary base pairing between the bases. And that complementary base pairing, top exam tip for you, always say what the complementary base pairing is. So A with T and C with G. So always say what the complementary base pairing is. And this complementary base pairing, this means that if one base is known, then the other base, the base for the other strand will know what the other base is as well, okay? What is it that forms between these complementary base pairs? So complementary base pairing between bases, what forms between the bases? What is it that forms between the bases? Nice there, Zara. Yes, working super hard today, Zara. I love it. Fantastic. Well done. It is those hydrogen bonds. So we've got our hydrogen bonds between the bases as well. And so our final mark here is the idea that if one base is known, it can pair with only one other base. So the base of one strand can pair with only one base on the other strand. And later on in year 12, we will go through this process of DNA replication in a bit more detail to really fully explain how the new strand of DNA gets produced, okay? About those 4 bases, so the 5 of the Moonlight, so adenine, thymine, guanine, cytosine, and then uracil for RNA as well. So that's what you need to know. Last question then before we finish today. So a group of students carried out an investigation to extract and purify DNA from a plant sample. They followed these steps. Combine the plant material with detergent, add salt, add protease enzymes, wind the DNA precipitate onto a glass rod. Suggest whether this procedure would be effective in extracting and purifying the DNA and justifying your answer. So what do we think? Would it work or not? Would it work or would it not? Would you be able to do this? Nice there, Zara. Yeah, so no mention of heating. So we could say that no, it's not going to work. Temperature too low. Enzyme activity wouldn't, not enough enzyme activity. So temperature too low. So enzyme activity too low. We need to add the ethanol. Good there, Moonlight. No ethanol added. How many marks would you get if you said that? So Zara, that would get you 1 mark here. So this would get you 1 mark. No ethanol added, so the DNA wouldn't precipitate. Remember, we need to justify our answer. No ethanol added, so the DNA won't precipitate. That would be another mark as well. They've added the detergent and they've said add the plant material, but what have they not done? Oh, Moonlight, you're jumping ahead of me. Fantastic. They have not homogenized the plant material. So no homogenization. And so the cells are not broken open. So the cells are not broken open. And again, that would get you a mark there too. Fantastic there, team. Okay, so have you enjoyed the lesson? Be honest.